All right, welcome to the Cell Division Challenge. Today, we're diving into one of the most essential processes in all of biology. But instead of a lecture, we're going to make it a quiz. I'll throw out the questions and you see how you do. Ready? Let's get started.
So, how's this going to work? Simple. I'll show you some key ideas about cell division with a few blanks. You take a crack at filling them in, and then we'll go through the answers together. It's a great way to test what you already know and maybe learn something new.
Okay, round one is all about the fundamentals, the building blocks. Before a cell can even think about dividing, we gotta know the key players involved, especially those all-important chromosomes that carry our genetic blueprint.
Here we go, first three questions. Super basic stuff to get us warmed up. What are all living things made of? What are the two main ways cells split? And where do we find our genes hiding? These set the stage for everything else.
And the answers are cells, meiotic division, and the chromosome. Simple enough, right? So, all life is made of cells, and those cells divide through either mitosis or meiosis, we'll get into the difference soon, and the chromosome, well, that's the star of our show. All right, let's zoom in a little bit on the chromosome itself. It's not just a single little strand, especially when it's ready to divide. What are its different parts called? And what's that crucial little point that holds it all together? Okay, the answers you were looking for are chromatids, sister chromatids, and the centromere. So when a chromosome copies itself, you get two identical arms, those are the sister chromatids, and they're joined together at that central spot, the centromere. And the location of that centromere is actually how we classify different chromosomes. If it's right in the middle, we call it metacentric. If it's way off to one side, it's acrocentric. And if it's right at the very tip, it's called telocentric. This structure is super important for how they get pulled apart later. And just like that, we're on to round two, the mitosis marathon. Now, mitosis is the process our bodies use for pretty much everything, growing, healing, you name it. It's all about making two perfect, identical copies from one original cell. Let's start with the big picture of mitosis. When does it happen? How do the new cells compare to the original one? And what are the two main events that make up the whole division process?
So, the answers are, after fertilization, the daughter cells are identical to the parent cell, and the process includes cytokinesis. You gotta have both parts. Karyokinesis, which is the nucleus dividing, and then cytokinesis, which is the rest of the cell splitting. You need both to get two new working cells.
But wait, a cell can't just divide on a whim. There's a whole prep phase it has to go through first. What do we call this period? And what are the crucial things that happen? Like, copying all the DNA before the main event. That crucial prep stage is called interphase, and a really important part of it is the synthesis phase, or S phase. That's when the cell makes a copy of its DNA, creating those double-stranded chromosomes. Before that, it's busy gathering up all the energy and proteins it needs.
Okay, once all the prep work in interphase is done, mitosis itself kicks off. It happens in four main stages: prophase, metaphase, anaphase, and telophase. A good way to remember the order is just PMAT. PMAT. Let's see what happens in each one. So let's look at those first two stages. In prophase, the cell is getting its copied chromosomes organized. What happens to the wall of the nucleus during this time? And then, in metaphase, where do all those chromosomes line up so perfectly?
So, in early prophase, the chromosomes are condensing, but they're hard to tell apart just by observation. The nuclear membrane starts to get disorganized and breaks down. Then, in metaphase, boom, they all line up right along the cell's equator, getting ready for the big split. Okay, this next one's a little trickier, putting it all together. What do we call the process when the chromosomes finally break apart? What happens to the amount of DNA after that split? Can you spot the missing stage in the sequence? And, this is the big one, what's the final chromosome count in the new cells? And the answers are karyokinesis for the chromosome breakage, the DNA content goes down to 2C, the missing stage is metaphase, and the final chromosome count is 2N equals 6. That last point is the whole takeaway of mitosis. The new cells are perfect genetic copies of the parent cell. Nothing's lost. Whew, okay, take a deep breath. We've made it to the final and definitely the trickiest round, the meiosis gauntlet. So if mitosis was about making identical copies, meiosis is totally different. It's about creating genetically unique cells, like sperm and eggs, for sexual reproduction. This is where genetic diversity comes from. All right, meiosis actually has two parts. Let's look at meiosis 1, where things really get different from mitosis.
Where do the chromosomes line up? What's the special name for how they separate? And what does this do to the cell's total number of chromosomes?
The answers are they line up at the equator, they're separated by disjunction, and the new cells become haploid. That process, disjunction, is the secret sauce of meiosis I. Instead of splitting a chromosome in half, it separates entire pairs of chromosomes, which is how it cuts the total number in half.
We're so close to the finish line. Let's wrap this up. What happens at the very end of meiosis I, and then what happens in meiosis II? You'll notice meiosis II looks a whole lot like mitosis, by the way. What are its key stages?
And our final answers are, in telophase I, the nuclear membrane reappears, creating 2 new cells. The DNA content is 2C. Then, in meiosis II, the chromosomes line up in metaphase II and get pulled apart in anaphase II. And just like that, you have 4 unique haploid cells. Great job! You made it through!
Wow, that was a lightning-fast trip through cell division. Doesn't matter if you aced it or learned a few things along the way, the point was to see the beautiful, logical steps in this whole process. So, let's just quickly recap the big ideas. So what's the big takeaway here? Well, it really boils down to 2 things. Mitosis is like the body's photocopier. It makes identical cells so you can grow and heal. Meiosis, on the other hand, is the genetic shuffler. It creates unique cells that drive the incredible diversity of life. Every living thing you can think of, including us, is a product of these 2 amazing, elegant processes. It's not just some trivia, it's the engine of life itself. Thanks for taking on the challenge with me.